Mitochondria, the powerhouse of the cell. I know, and I think all of you know, that that is a huge oversimplification. But there is something else important about mitochondria that I didn't know, and I'd like to show it to you. Sure, they burn glucose to produce our ATP, but they also make a lot of other molecules, such as the bases of our DNA. They send signals to the rest of the cell. For example, if the oxygen levels get too high or too low, the mitochondrion can signal the external cell to begin cell death, apoptosis. And the mitochondria know everything about oxygen. All of the reactive oxygen operations are primarily concentrated here, away from the rest of the cell, because they can do great damage. I've shown Mr. Yuck playing the role of a peroxide here, a normally occurring exhaust product and signaling molecule, not very dangerous, at least until an iron atom drifts by. This is the most dangerous and reactive radical in all of earthly biology, the hydroxyl radical. It only lasts for nanoseconds, but whatever it touches will be badly damaged. Mitochondria make power, mitochondria make mistakes. So some of the amino acids are manufactured here. And they don't just burn glucose, they burn the amino acids from protein used as fuel. They burn fatty acids too. Everything is used to make ATP and to move electrons and ions to the right places. They burn so much, carbs, fats, and protein, that calling them the powerhouse isn't wrong, as long as we remember it's just part of the story. And interestingly, for our story, they have their own DNA in a little ring. So that's my cartoon Mito with lots of inputs and outputs. Most of this is review for many of you, but if not, the impression that it's not just a powerhouse is a good one to make. And I still haven't told you the one component of the mitochondrion that I didn't know about or why I didn't know about it. Let me detour for a moment and let's discuss ribosomes. I just heard many of y'all say in unison, the protein factories of the cell. Well, that's not an oversimplification. That's fundamentally correct. And I ask you, where are the ribosomes? And most of you say they're the little round black spheres dotting the folds of the endoplasmic reticulum. And that's where I was missing something. I had read an article describing research into the evolution of the ribosome in animals. Now, a ribosome is made of dozens of proteins, but at the very core is a piece of RNA, not DNA, RNA. It's the peptidyl-transferase center, the PTC. And that piece is ancient. It's older than LUCA. It's probably older than DNA. It's probably related to the original proto-life, the original RNA world, where it was a system that put together amino acids into more useful, longer peptides. Unfortunately, the article was back at the powerhouse of the cell. So I found the original paper, found the preprint. The paper was behind a paywall. The PTC teaches us profound lessons about the entire tree of life and the origin of life itself. And here it was shedding light specifically on animal lineages, and it was fascinating, but it was a little difficult. I have zero formal training in biochemistry. I have to keep a cheat sheet of acronyms. But then one section didn't make any sense. It was suddenly talking about an older, simpler system of ribosomes with a little bit different PTC. And that's exactly what I was missing. Mitochondria have their own ribosomes. Think about the origin story of mitochondria. The classic story is that an alpha proteobacterium got engulfed by an archaeal cell, except it most probably wasn't a single occurrence where one isolated cell got engulfed by one other cell. Some of the closest relatives of the cells that became mitochondria are parasites. They live inside of other cells, stealing nutrients. So it's likely a population of these parasites living inside of a population of large, complicated cells, and they're all making bacterial whoopee in lots of complicated ways. The engulfed bacteria have a full genome, but as they become mitochondria, they lose a lot of their genes, and a lot of their genes move to the chromosomes in the cell's nucleus. They get left with just 37 genes. That seems like an understandable number, doesn't it? A few proteins, a lot of transfer RNAs, and those 37 are highly conserved across all complex cells and nearly perfectly conserved across multicellular animals. Along with all the other features of the cell, the DNA, the membranes, the bacteria that got engulfed had their own ribosomes, their own protein factories, their own version of the PTC inside those ribosomes. That makes sense, right? And that's what I was missing, trying to understand the incredible paper I was reading. Mitochondria also have ribosomes in addition to the cell's ribosomes in the endoplasmic reticulum and a few scattered elsewhere. Those ribosomes are much simpler than the main ribosomes. The RNA core at the center is closely related to bacteria and gets built from the mitochondrial DNA. So now it makes sense. We can track the evolution of animals from the PTC and the main cellular ribosomes. We can also track it in the PTC of the mitochondrial ribosomes. This has some impact in medicine too. Several diseases are caused by malfunctions in your mitochondria. Some other diseases are caused by malfunctions in your ribosomes. And it turns out that there are some terrible diseases caused by malfunctions of the mitochondrial ribosomes, mainly in infants. They're being researched right now by some brilliant biochemists. And if any of you happen to be in the channel right now, part of my gentle audience that studies this topic, please leave us a comment. I'd love to send you a channel t-shirt. Mitochondrial ribosomes, that's probably not the most interesting topic we could cover about mitochondria. For instance, mitochondria also have the highest voltage gradient in all of earthly biochemistry across their membranes. It's much higher than electric eels. It's even higher than the voltage differential that triggers lightning. Learning that mitochondria also have ribosomes just made the story click into place for me personally. I'd never heard that before, and I read and watch a lot of science journalism. That's the level of knowledge that actual researchers know and use every day, but it doesn't get explained to those of us outside their field. The specific, esoteric parts of the knowledge base that make it possible to fully understand the big picture without stretched analogies just happen to be the most fun parts. Let's find some more of those right after this. We're looking for fun science that actual researchers know and use every day, but it doesn't get presented to us outside of their field. Sometimes that means learning their tools. Let's look at currents in the ocean. A lot of you are already pointing at this place, where the cold, very salty water off the coast of Greenland dives down, the place that drives the major ocean circulation for the whole world. That's not today's topic. I promise we'll revisit it. But spoiler alert, the AMOC stopping is a serious tipping point, but it's only the third worst that we'll cover. On the basin scale, say the size of an ocean, the Gulf Stream takes a few months for a drop of water to go from Florida to Europe. The core of the Gulf Stream is the fastest sustained current in our oceans. There are other currents up here at the basin scale, like currents driven by trade winds, but they're all relatively slow, slower than a river you can stand beside. Let's go down in the size scale, down to the Meso scale. This map is actual surface current data from satellites. The eddies are in the mesoscale size range, typically 10 to 100 kilometers across. Meso, from the Greek root meaning awkwardly in between things. The eddies last a few months typically, and they have fairly small velocities. They're pretty easy to model on the scale that the big climate simulations use. Interestingly, the ones that loop clockwise are quite different from the counterclockwise swirls. I'll come back to that in a minute. They look very dramatic, but they're not the biggest movers or actors. So let's drop down even further in size, down to the sub-meso scale. Our features down here are between 100 meters and 10 kilometers. And down here, a unit of water can be very different from the unit next to it. Vertical speeds might be tens to hundreds of meters per day. The features down here are typically fronts, thin walls between two different masses of water that might have very different temperatures or salinities or especially velocities. And we have the energy down here to really mix things up. We can punch down through a thermocline here. Now, how fast, how much energy? I can give you meters per second or gallons per minute or gigajoules. If we were actual oceanographers, we would talk about flow in a unit of measurement called a Sverdrup. But in science, one of the best ways to handle topics like this is with a dimensionless number. This is a quantity or a ratio where all of the units drop out, cancel out, leaving us with a pure number. If you were a radio engineer or just a stereo nut, you would talk about signal to noise, decibels over decibels. If I tell you that a pilot experienced 3 Gs, you would know what he felt. I bet that you have several dimensionless numbers in your field that really make a lot of sense. But the number we're going to use in these ocean currents is the Rossby number. What this means in words is that the Rossby number is the ratio of inertial forces to Coriolis forces. I had never encountered this term anywhere in a lifetime of reading and watching science, and yet oceanographers and weather analysts, they know this better than I know my car's tire pressure. At some scale, this dominates weather, such as the strength and speed of the jet stream. Here in the North Atlantic, our little sub-meso scale eddies and fronts, they have very large Rossby numbers. They are not dominated by Coriolis forces. Instead, they're dominated by a little more powerful local conditions, temperature, pressure, velocity. These features are strong enough to push CO2 down far enough to be captured or to bring nutrients up for an algae bloom. These features are the ones that cause the little quick local surface temperature changes that can kill a hurricane or intensify it. The scale of these features is just too small to be captured reliably by the big climate simulations and too chaotic to be simply averaged out. Sub-mesoscale fronts. They are not quite as fast or as powerful as a tuna fish, but they dominate the tuna's home. The other eddies that we saw at the mesoscale, they're big, they're slow, they have very small Rossby numbers. They are dominated by Coriolis forces. In fact, in the counterclockwise ones, the water at the edge is being pulled away by Coriolis forces, and that brings water and nutrients up to fill the gap. Meanwhile, the clockwise features are bringing oxygen and water down. So Rossby numbers, inertial over Coriolis, they immediately give us the answers to these behaviors. This is YouTube, and I'm making jokes about units of measurement. I haven't blown anything up. So, what happens if you heat up a magnet? If you heat up a magnet near its Curie temperature, it quits being a magnet. But the Earth's inner core is a solid piece of nickel iron with a whole lot of gold and a whole lot of platinum, but it's thousands of degrees. It can't be a magnet. Instead, the outer core is liquid iron nickel, and it's highly conductive, and it's moving. Convection cells, moving heat from the inner core to the mantle, move big columns up and down about the speed of a snail. The molten metal is a great conductor. If there's even a tiny bit of magnetic field at the beginning, even just the galactic background, then we've got moving conductor in a magnetic field, which generates electrical currents, which generate more magnetic field, and the system becomes a self-exciting dynamo. The white hot, solid inner core isn't a magnet at all, even though it is a great conductor and it responds to the dynamo in the outer core. Inside the sun, roughly the same thing happens, but in a different place and with even wilder, crazier shapes. Hydrogen is an insulator at standard temperature and pressure, but in the sun, it has been completely ionized into a plasma. Both the electrons and the positively charged nuclei freely conduct current. So again, we've got a moving conductor in giant convection cells makes another big dynamo. It is a ginormous thermonuclear bomb bouncing around on an 11-year cycle, but I'm going to call the ionized plasma normal as I swap to this picture. The magnetic field of Jupiter is very different, lopsided, tilted, and ferociously strong. You're familiar with the Great Red Spot, but when we look at the magnetic field, there is a huge feature near the equator called the Great Blue Spot that behaves almost like a second South Pole. Even ignoring that, what makes the conductor that makes the magnetic field in Jupiter is a quantum phenomena, and it's a really strange quantum. Deep inside Jupiter, the temperature isn't very high, but the pressure is. You've probably heard of metallic hydrogen, but have you ever read a good description of what its substance is like? Because that's pretty rare outside of a condensed matter physics journal. For one thing, in Jupiter, the metallic hydrogen is liquid. It's not a metal like we're used to. So this isn't like in the sun, where the electrons get stripped off by the high temperature and they're ionized. Instead, the hydrogen has entered a completely different quantum state. It has undergone a Mott transition. The nuclei get squeezed very tightly together. In fact, if the electrons had remained in their original orbitals, then the spacing of those orbitals would be very small compared to the size of the orbitals. They're all overlapping with their neighbors. The electrons overlap so completely that they lose their identity and the wave functions smear together. The electrons delocalize into one big giant quantum sea. Below a certain level, metallic hydrogen is about as runny as water and very conductive. So we get big convection cells, we get the movement of conductors, we get the original magnetic field, and once again, a great big dynamo. I don't know, do you guys like analogies? Iron, a lot of electrons. It can't hold on to the outer two because they're so far away, so they fall off. In the sun, temperatures are so high, the electrons get burned off of the atoms. But for metallic hydrogen, the nuclei are pressed so close together that the orbitals almost completely overlap, and the best analogy is smoosh. If you're like me, you read and watch and devour a lot of science, and sometimes the analogies just aren't enough. Later in my education, I took a class in the failure of materials, how metals broke, instead of using the approximations. I remember feeling distinctly angry, thinking, why didn't they teach us this earlier? This stuff is important. And I'm sure that you felt that way about a topic in your field. So there are viewers among you right now who are doctors and biochemists, and you already knew about mitochondrial ribosomes. There are among you people who have solved Schrodinger's equation. Now, the Venn diagram for those two groups of people, I wouldn't tell a molecular biologist a story about the powerhouse of the cell. So why not let's let the oceanographers in on the better description too? If you've enjoyed these little bits of science, then please hit like. It really helps us a lot. If you want to see another episode, then please subscribe. You figure out the bell thing. But even better than that, comment below. Point out my mistakes. Get in huge flame wars in the comments about which parts I exaggerated. That helps tremendously. I read as many comments as I can, and I'll respond often. But the absolute best thing that you can possibly do for us is to leave a comment about a topic from your field. Suggest a topic that I should research and present, something you know is important, fascinating, and never explained well in typical science journalism. We're heading somewhere with this channel. The trip there will be interesting, and the destination is crucial. I think I'll be able to convince you of that. Until the next episode, please be kind to everybody, and let's keep being scientists, all of us.